Hello everyone, I am excited to present the work we have been developing, which is part of the larger initiative Operación Esperanza. My name is Tania Valdivia and I am a postdoctoral researcher at the CIFNOR in the Baja California Peninsula of Mexico. Today, I'll share the vaquita eDNA project objectives, methods, and findings as we strive to contribute to the conservation of this species. The vaquita porpoise is the world's most endangered marine mammal, endemic to the upper Gulf of California. With fewer than 10 individuals remaining, it's on the brink of extinction, primarily due to bycatch in illegal gill nets. Traditional monitoring methods such as visual surveys and acoustic monitoring are limited in effectiveness due to the vaquita's elusive nature and the challenges of the dynamic environment. eDNA-based methods offer a promising alternative for vaquita research because it can improve our understanding of vaquita distribution. However, challenges remain, including environmental factors in the upper Gulf of California that can affect eDNA degradation and detection. Addressing these issues is essential to maximizing the effectiveness of eDNA monitoring and enhancing conservation efforts for the vaquita. To date, no qPCR assays exist specifically for the vaquita porpoise. However, various global studies have successfully utilized species-specific eDNA methods to detect the rarest marine mammals. This represents a significant opportunity to apply eDNA techniques to the vaquita research. Our objectives included designing species-specific primer pairs for detecting the vaquita porpoise using qPCR and ddPCR, testing the sensitivity and specificity of the assays, and sampling marine eDNA to evaluate the assay's performance in real-world conditions. This project has been developed in two main phases. The first phase was a pilot project conducted in 2022, where we tested the viability of detecting vaquita eDNA and designate initial qPCR assays. The second phase is the Operation Esperanza project, currently underway in 2024. This phase builds upon the findings from the pilot, aiming to refine our methodologies, validate our assays, and enhance our ability to detect the vaquita effectively. To design primers specific to the vaquita porpoise, we utilized 12 vaquita mitogenome sequences and 34 mitogenomes from marine mammal species, including 18 that co-distribute with the vaquita. We confirmed a 100% match with the vaquita sequences using primer blast, ensuring there was no overlap with non-target species. This validation process was essential for developing reliable primers for detecting vaquita eDNA. In the cross-amplification test, we assessed the specificity of our selected primer pairs using DNA from co-distributed marine mammals. Utilizing these negative control samples, we performed qPCR followed by MEL curve analysis to confirm the specificity of the amplification and to evaluate primer consistency. While some primers exhibited non-specific amplification, others demonstrated high specificity, amplifying only vaquita DNA with distinct MEL curve profile. In 2022, we visited 10 sites in the zero-tolerance area of the vaquita refuge to collect eDNA samples. To prevent filter clogging from sediment, we used a pre-filtration step using a 1.2 micron filter to remove larger sediment particles, followed by filtration with 0.45 micron filter. This sampling scheme allows us to collect sufficient eDNA to effectively validate our assays and assess their performance in the field. Our sampling yielded eDNA to test the detection assays for the vaquita porpoise. Results showed that 4% of the qPCR reaction returned positive detections for vaquita eDNA. Of these, 7 detections were confirmed through Sanger sequencing, providing direct evidence of vaquita presence. The highest detection frequency was observed at sites 18 and 14, with the majority of positive samples coming from the 1.2 micron filtered seawater. During our pilot project, we tested 10 primer pairs and successfully identified candidate detection assays with good amplification efficiencies and high sensitivity and specificity. However, we also encountered several challenges that impacted our sampling efforts. Therefore, derived from the pilot project outcomes, the recommendations for the 2024 phase were to prioritize methodological refinement, implement onboard sampling filtration, and investigate eDNA transport and decay dynamics. In 2024, during the May survey occurring along with the visual and acoustic surveys, we implemented an on-site filtration protocol using a 5-micron self-preserving filter and the Smith root system, allowing us to filter seawater directly on-site efficiently. This allows us to conduct extensive sampling of the zero-tolerance area in the Vaquita Refuge, collecting 387 liters of seawater. Additionally, we collected opportunistic samples during Vaquita sightings and filtered water for eDNA decay and transport evaluation. We also gathered samples about 200 meters from sightings to potentially gain insight into species genetic diversity. The next steps involve a comprehensive analysis of the eDNA samples collected during the 2024 sampling campaign, including eDNA extraction and qPCR or ddPCR amplifications to evaluate the presence of Vaquita DNA in the filtered samples. We will also complete the analysis of eDNA degradation and transport experiments based on samples collected near Vaquita sightings. Validation will also continue for new assays, involving sensitivity and specificity tests using positive and negative controls. Transfer of the detection assays to ddPCR equipment to compare the sensitivity of the detections. Once the analyses are complete, we will compile our findings into technical reports and publications, sharing them to the scientific community and stakeholders involved in conservation efforts. Finally, I would like to thank the Operación Esperanza team and the pilot project team, the collaborators, and the funding partners for their support. I appreciate your attention. Please get in touch if you have further comments or questions.